The incredible diversity of our terrestrial biosphere is rooted in an evolutionary journey stretching back over 3.5 billion years. In these ancient oceans, the first photosynthetic organisms emerged, cyanobacteria that would forever change our planet. These bacteria performed the first oxygenic photosynthesis over 3.4 billion years ago. Chemical signatures in ancient rocks and stromatolites confirm these early photosynthesizers existed at least 3.4 billion years ago. Their oxygen-producing capacity triggered the Great Oxygenation Event about 2.4 billion years ago, fundamentally altering Earth's atmosphere. This set the stage for aerobic metabolism and the rise of complex life. The origin of chloroplasts occurred through endosymbiosis of cyanobacteria by early eukaryotic cells. This created the first photosynthetic eukaryotes, with modern plants inheriting chloroplasts from green algal ancestors. Multicellular green algae emerged in Proterozoic times up to 1 billion years ago. The most ancient carbon fixation pathway is C3 photosynthesis, present in nearly all photosynthetic lineages. As atmospheric CO2 decreased and oxygen increased, C4 photosynthesis evolved independently over 60 times. CAM photosynthesis evolved as an adaptation to arid environments, allowing plants to open stomata at night to minimize water loss. The shift from water to land represents one of Earth's greatest evolutionary leaps, occurring nearly 500 million years ago. Key innovations included protective spores with sporopollenin walls and waxy cuticles to prevent water loss. Fossils from the Rhynie chert reveal these early terrestrial communities from 410 million years ago. These early plants developed alternation of generations and retained embryos within maternal tissues for protection. Symbiotic associations with mycorrhizal fungi were crucial for early land plant success. These partnerships improved water and nutrient uptake, with fossil evidence dating back over 400 million years. This ancient mutualism enabled the colonization of nutrient-poor terrestrial environments. The first vascular plants like Cooksonia appeared in the Silurian period, 430 million years ago. Vascular tissues, xylem for water transport and phloem for nutrients, enabled larger plant size and true terrestrialization. These innovations led to the development of the first forests and complex terrestrial ecosystems. Plant photosynthesis continued to shape Earth's atmosphere and climate throughout their evolution. Plants became key drivers of global biogeochemical cycles, influencing carbon and oxygen levels. Their impact on atmospheric composition set the stage for all subsequent evolution. From the Archean emergence of cyanobacteria to the Ordovician land invasion, each geological period brought new innovations. The fossil record provides a detailed chronicle of plant evolutionary milestones across deep time. Exceptional fossil sites like the Rhynie chert offer unprecedented windows into ancient plant communities. Changing climates, atmospheric composition, and geological processes drove plant evolutionary innovations. Plants faced extreme environmental pressures including UV radiation, temperature fluctuations, and chemical stress. Each challenge drove the evolution of new protective mechanisms and survival strategies. Modern molecular phylogenetics has revolutionized our understanding of plant evolutionary relationships. Molecular clocks calibrated with fossil evidence reveal the timing of major evolutionary events. Genomic studies detail the gene duplications and innovations underlying plant evolutionary success. These early evolutionary innovations laid the foundation for the complex plant communities we see today. From simple cyanobacteria to complex multicellular plants, each innovation built upon previous adaptations. This evolutionary journey set the stage for the remarkable diversification that would follow. The transition from simple photosynthetic bacteria to complex land plants represents one of evolution's greatest achievements. These early innovations in photosynthesis and terrestrialization transformed our planet into the green world we know today. Understanding this deep history provides crucial insights into how plants conquered land and shaped the biosphere. But this was only the beginning of an even more remarkable story of innovation and diversification. The evolution of vascular tissues, roots, leaves, and seeds would revolutionize plant life even further. These innovations would lead to the rise of forests, the evolution of seeds, and ultimately the flowering plants that dominate today. The stage was now set for the next great leap in plant evolution. The evolution of vascular tissues, xylem and phloem, represents a landmark adaptation enabling increased plant size. Fossil taxa like Cooksonia from the Silurian period mark the first clear evidence of these transport tissues. These systems evolved from simple conducting strands to complex specialized cell networks. Secondary growth, regulated by the vascular cambium, enabled the formation of wood and the rise of trees. This innovation led to the formation of the first extensive forest during the Carboniferous period. Lignified tissues provided the mechanical support necessary for plants to reach unprecedented heights. Roots evolved as specialized organs for absorption and anchoring, initially from simple rhizoid systems. True roots with apical meristems and vascular connections arose in the late Silurian to Devonian periods. Root evolution occurred independently in lycophytes and euphylophytes, demonstrating convergent evolution. Leaves evolved multiple times, microphylls in lycophytes and megaphylls in ferns and seed plants. Megaphylls with complex branching veins dramatically boosted photosynthetic efficiency. These innovations played a major role in the cooling and oxygenation of Earth's atmosphere through enhanced photosynthesis. Seeds evolved from heterosporous ancestors producing separate microspores and megaspores in the late Devonian. Seeds included protective integuments, nutrient-rich tissue, and mechanisms for dormancy and dispersal. This innovation liberated plants from dependence on water for reproduction. Seeds allowed colonization of drier and more variable environments, representing one of plant evolution's most consequential innovations. Gymnosperms, the naked-seeded plants, diverged in the late Devonian and flourished through the Mesozoic. The main gymnosperm groups, conifers, cycads, ginkgo, and natales, show remarkable diversity in form and ecology. These plants dominated terrestrial ecosystems for over 200 million years. Flowers, unique to angiosperms, first appeared in the late Jurassic to early Cretaceous, 150 to 125 million years ago. Flowers are condensed shoots with sterile and fertile organs organized in whorls, controlled by the ABCE gene network. The interplay between flower structure and animal pollinators triggered a spectacular evolutionary radiation. Angiosperms rapidly diversified and became globally dominant, comprising 90% of living plant species today. Key innovations included efficient vascular systems, rapid growth rates, and intricate ecological interactions. Double fertilization and fruit development provided additional advantages for seed protection and dispersal. Bryophytes, mosses, liverworts, and hornworts represent the earliest land plants, retaining many ancestral features. They maintain a dominant gametophyte generation and require water for fertilization. Despite their simplicity, bryophytes continue to diversify and occupy specialized ecological niches. Ferns underwent dramatic radiations during the Paleozoic and experienced a renaissance in angiosperm-dominated ecosystems. Modern ferns retain both homosporous and heterosporous reproductive strategies and show evidence of genome duplications. Tree ferns demonstrate that non-seed plants can still achieve remarkable size and ecological prominence. Lycophytes were among the first tree-sized plants dominating Carboniferous forests as giant club mosses. Today's lycophytes are much smaller but retain the basic body plan of their ancient relatives. Their massive Carboniferous forests formed much of the coal we use today. Each major innovation, vascular tissues, roots, leaves, seeds, and flowers, built upon previous adaptations. This stepwise accumulation of innovations enabled plants to exploit new environments and ecological opportunities. These innovations didn't just change plants, they transformed entire ecosystems and global biogeochemical cycles. Genetic mechanisms including gene duplications and regulatory changes provided the raw material for innovation. Regulatory networks like the ABCE genes controlling flower development demonstrate how genetic innovation drives morphological change. Whole genome duplications created opportunities for evolutionary experimentation and stress tolerance. These fundamental innovations set the stage for the incredible diversification we see in modern plant communities. But innovation alone wasn't enough. Environmental factors and ecological interactions shaped how these innovations were expressed. Climate, soils, and biological interactions determined which innovations succeeded in different environments. Understanding these environmental drivers reveals how plants became the dominant force shaping terrestrial life. Earth's climatic evolution powerfully influenced plant evolutionary trajectories throughout deep time. Warm, humid periods facilitated the rise of coal forests and fern dominance. Arid intervals favored gymnosperms and later C4 and CAM photosynthesis in angiosperms. Changing atmospheric CO2 and oxygen directly affected plant physiology and photosynthetic pathway evolution. The evolution of C4 photosynthesis in grasses was a direct response to declining atmospheric CO2 in the Cenozoic. High oxygen levels made C3 photosynthesis less efficient, driving the evolution of carbon-concentrating mechanisms. Early land plants and their mycorrhizal partners initiated weathering of rocks, generating the first soils. Plants established biogeochemical cycles that continue to shape terrestrial ecosystems today. Root architecture evolved to respond dynamically to soil nutrient availability. Mycorrhizal associations, nearly universal among land plants, have ancient evolutionary roots. Arbuscular mycorrhizal fungi are recorded in fossils over 400 million years old, as ancient as land plants themselves. Nitrogen-fixing symbioses provided competitive advantages in nutrient-poor soils and evolved multiple times independently. Animal-plant coevolution, especially with pollinators, drove rapid radiations in flowering plants. Seed dispersal mutualisms allowed plants to colonize new habitats and reduce competition with parent plants. Plant-herbivore interactions drove the evolution of complex chemical and physical defense systems. These interactions created evolutionary arms races that increased biodiversity in both plant and animal lineages. Adaptive radiation filled ecological niches created by key innovations, climate change, or competitor extinction. The spectacular angiosperm explosion in the Cretaceous exemplifies rapid adaptive radiation. Plants evolved to exploit virtually every available terrestrial and aquatic niche. Gene and whole genome duplications generated raw material for evolutionary novelty and stress tolerance. Hybridization and introgression, especially common in plants, facilitated rapid adaptation and speciation. Regulatory changes in developmental genes allowed the same genetic toolkit to produce remarkable morphological diversity. Today, plants comprise over 300,000 described species, with angiosperms representing 90% of this diversity. Plants provide essential ecosystem services including oxygen production, carbon sequestration, and climate regulation. Human agriculture has created new selective pressures, leading to the evolution of crop plants. Today, plant diversity faces unprecedented threats from habitat destruction and climate change. Many plant species are becoming extinct before we can fully understand their evolutionary history and ecological roles. Rapid climate change is forcing plants to adapt or migrate at unprecedented rates. Genomic and computational advances promise even deeper insights into plant evolutionary mechanisms. Phylogenomic studies using thousands of genes are revolutionizing our understanding of plant relationships. Experimental evolution studies allow us to observe evolutionary processes in real time. Learning from plant evolutionary history reveals both the resilience and vulnerability of life. Plant innovations inspire technological solutions to human challenges. Understanding plant evolution informs conservation strategies and sustainable development. The history of plant evolution over geological time reveals relentless innovation and environmental adaptation. Each innovation built upon previous adaptations, creating the complex tapestry of modern plant life. Plants have been both architects and engineers of the terrestrial biosphere. Understanding this journey requires integrating fossils, genetics, biogeochemistry, and ecology. Plant evolution continues today as species adapt to changing environments and human influences. As humanity faces rapid environmental change, learning from plant evolutionary history is more relevant than ever. From simple cyanobacteria to complex flowering plants, this evolutionary journey transformed our planet. This remarkable story of innovation, adaptation, and diversification created the green world that sustains all terrestrial life. Understanding plant evolution provides crucial insights for conservation and sustainable stewardship of our living planet. The evolution of plants, a testament to life's creativity, resilience, and transformative power across deep time.